Hi, this is Imani Canton,
Hi, I'm Kiera Archer.
Hi, I'm Dina Gibson.
and this is our MSH2 midterm presentation. This is our abstract outlining the background, methods, and results of the experiment. MSH2 is a member of a set of genes known as mismatch repair genes. MSH2 codes for a protein that plays a primary role in DNA repair. However, mutations in the MSH2 gene can occur and cause Lynch syndrome, also known as hereditary non-polyposis colorectal cancer. MSH2 gene mutations can cause the production of an inactive MSH2 protein and thus interfere with DNA repair. When MSH2 is disturbed, the amount of mistakes in DNA left unrepaired will accumulate and cause the formation of a tumor. The goal of this project is to determine whether or not repair is the same or worse than wild-type MSH2. Human SNP is engineered into yeast MSH2 gene. Engineered plasmid introduced into DNA into E. coli because it produces more plasmid DNA for experimentation. A plasmid mini-prep is carried out to extract DNA. Gel electrophoresis, PCR, and restriction digestion are used to identify the three plasmid DNAs, PMSH2, PMSH2', and PRS413. SP1 and SP4 are the same size DNA, therefore are PMSH2 and PMSH2'. Restriction enzyme BST-Y1 is used to cut the MSH2 in order to distinguish between wild-type and mutant MSH2.
Okay, now here's the introduction. And in the introduction, I'm just going to give a little bit more information about Lynch syndrome. Cancer or Lynch syndrome is caused by a heterozygous mutation in the mismatch repair gene. And Lynch disease or Lynch syndrome is caused by point mutations that occur within various MSH2 genes or different variations of those particular genes. And individuals who suffer from HNPCC are at increased risk of colorectal cancers and other aggressive forms of cancer such as uterine cancer, ovarian cancer, liver cancer, or kidney cancer. And some individuals may have mutations in a mismatch repair gene, but they will not develop cancer. And also it's important to say that anyone in any ethnicity can be prone to getting Lynch syndrome, and it usually occurs in people who are younger than the age of 50, because most of the time an individual will get colon cancer as they age, but this particular form of cancer caused by HNPCC is more common in people who are younger than the age of 50. And in our methods and materials, the MSH2 project, it consists of various lab experiments that are formed on a weekly basis. And each particular lab experiment for our project relates to a subject in molecular biology. Some examples of our experiments include plasmid mini-prep, restriction digestion of plasmid DNA, and polymerase chain reaction. The materials that are needed for the experiment vary depending on the type of experiment, but the use of the plasmid DNA extracted from E. coli strands on a weekly basis are used regularly. And it's also important that you have very great pipetting skills, because your values must be very accurate in order to get accurate results.
The experimental design for week 3, which is also our intro experiment, calls for us to isolate plasmid DNA from 3 E. coli strains using the plasmid mini-prep kit. And then we also were required to measure the concentration and quality of DNA in the samples using the nanodrop instrument, which is what this chart is explaining. Our results from week 3 show that SP0 had 330.5 nanograms per microliter and a 260 per 280 purity assessment of 1.84. SP1 had a concentration of 75.7 nanograms per microliter and a purity assessment of 1.88. And lastly, SP4 had a concentration of 140.6 nanograms per microliter and a 1.80 purity assessment. And our numbers are very close to the ideal number 1.9, which shows that our measurements were fairly accurate.
Week 4 began our gel electrophoresis and PCR experimentation. First, we need to dilute our samples from the chart that we just looked at to a 20 microliter reaction. This is the chart representing the 3 samples that we would include enzymes in and 3 samples that we would not include enzymes in to introduce into gel electrophoresis. Then we just needed to describe the samples. After adding the gel electrophoresis gel, loading gel, it was just a green appearance in all of them. As you can see, these 3 didn't have enzymes. Those are going to be our uncut, and then these 3 have a restriction enzyme. This would be our cut. And in this experimentation, we were using XBAI1 and APA1. This is our results from the first gel, and we just used this to distinguish, well, just to see the closed closed vector and the MSH2 wild type and the MSH2 mutant. We expected that, well, this is our this is our closed vector because it's lighter than the other 2, which means these are going to be our MSH2 and our wild type and our mutant MSH2.
The experimental design for week 5 called for us to actually carry out the process of PCR, or polymerase chain reaction, to amplify a region of the MSH2 gene that contains the mutation, which for our specific experiment is SP4.
Okay, this is a restriction enzyme analysis, and we're doing that for our wild type and our mutant strand of DNA, our plasmid DNA. And what we found was our restriction enzyme to be BSTY1. And as you can see, that is found only in our SP4 strand. And for this particular restriction enzyme, it will cut at 13 different areas within a DNA sequence, all at various size. So this is actually really a good restriction enzyme, because if there is any type of error or mutation that occurs within the sequence, this particular mutation has a great chance at removing it, because it cuts at so many different places at various sizes.
And the reason we needed to do the restriction enzyme analysis before this week, so that we can determine which a restriction enzyme to use.
And so the procedures for that were we had to digest the 3 PCR products using the restriction enzyme, which we identified as BSTY1, so that we can distinguish our mutant MSH2 from the wild type MSH2, and then actually run the digested products on the agarose gel to determine which plasmid DNA contains the MSH2 insert. And our specific enzyme, BSTY1, had an ideal temperature at 60 degrees Celsius, and the name of the reaction buffer was 10x NEB buffer 2.1. The experimental results are our results from the agarose gel run, and the first lane is our KB DNA ladder, and then the second one is uncut SP1, cut SP1, uncut SP0, cut SP0, uncut SP4, and cut SP4. And here we have a little side note that says the lanes are named incorrectly. SP0 and SP4 should be switched. So these right here should go over here in the middle. And Imani will explain a little further our results in the discussion.
So after gel electrophoresis, to determine the size of the plasmids, the mutant MSH2 and wild type MSH2 were the same size. You can go back. You can see that these are at the same level, if you want to think of it like that, and this one's a little lower. And this is expected because PRS413 is smaller than the mutant and wild type MSH2. That's the empty vector. And these results support the expectations for the size of the plasmids. In week 6, restriction enzymes were chosen using NEB cutter. BSTY1 was chosen to cut the plasmid because only the mutant MSH2 contained this restriction enzyme. Then gel electrophoresis was performed once again, so then we can distinguish between MSH2 mutant and the wild type. However, the gel was inconclusive because SP1 and SP4 appeared to be the same. Therefore, distinguishment was not possible. As you can see, they're all about the same. So in terms of determining which one is which is inconclusive. However, if we were to repeat this experiment again, there would be better micropipetting skills while doing gel electrophoresis in terms of loading into the dye and letting the dye stay the full time. In terms of further implications, we would further study different ethnicities and their rates of the mutation within the MSH2 gene. So we can look at African Americans versus Caucasians or any other ethnicities. We could also research other types of DNA repair genes that have point mutations that affect humans that can cause other diseases besides cancer. And we thank you for listening to our presentation and hope that you understood and learned about this MSH2.